Hi guys, and welcome to the first of the genetics videos. This one's going to look at genes and chromosomes. Firstly, we now know from our experiments and also from our theoretical work that the DNA is located in the nucleus of the cell. It's gathered in strips, long chains that are wound around proteins, and these long strips are called chromosomes. Now, chromosome lengths vary in their number and also in how long they are between different species. But the number of chromosomes is always an even number. And this is because chromosomes come in what we call homologous pairs. And homologous pairs are chromosomes where one has come from the mother and one has come from the father. In humans, there are 46 chromosomes, which is equivalent to 23 pairs. 23 that have come from your mum and 23 from your dad. We've already sort of overviewed the whole DNA, genes and chromosomes, but just for repetitive emphasis, here is the cell. The nucleus is inside the cell and there's a nuclear membrane around the nucleus. And inside that are these chromosomes. Generally speaking, they're wound tightly around proteins and we can't see them. But occasionally during the cell division processes, the chromosomes thicken and start to unravel a little bit as they are being copied, and so we can see them. When we see them, they look kind of like little X-shaped structures, but what we know is that they're actually very long. They've been coiled around a protein, so this section here. And when we stretch them out, we find this kind of a structure here in the section that shows you the gene. Now, this, as we've seen from our models, consists of sugars and phosphates alternating, and then coming off each of these sugars is a particular base. And those bases are either A's, T's, G's, and C's. And we use our Tony Abbott and George Clooney to memorize which ones go together. The gene then is a particular length of DNA and it codes for a particular characteristic. Now that's kind of the official definition. We're going to look at genes that are passed on from one generation to the next and that seem to code for specific characteristics such as whether or not you can roll your tongue. But we also know that genes also code for the production of particular proteins, such as we've talked about amylase, which is an enzyme that's present in your saliva and breaks down starch. Each individual chromosome may hold several hundred or even over 1,000 genes. Now, when we put all the chromosomes together, we get what we call a human karyotype. Now, the karyotype is what all of your chromosomes together looks like. You can see this particular karyotype has 46 chromosomes altogether, and an X and a Y, and so this would be a male. The chromosomes are made of DNA, and each of these contains genes in a particular linear order. And we know that on the basis of the sequence of bases that we've looked at, the A's, the T's, the G's, and the C's. We also know that the human body contains 46 pairs, and you can see there are 46 chromosomes, and you can see there are 23 pairs, 22, and then the 2 sex chromosomes. And these are the determinants of whether an individual is going to be a male or a female. If they're 2 X's, then it's going to be female. And if they are an X and a Y, then it's going to be a male. The chromosome pairs 1 to 22 are called autosomes. These are the ones that don't carry any characteristics that tend to relate specifically to the gender or sex of an individual. Now, this doesn't just happen to correspond to sexual characteristics. It can also correspond to certain things that seem to be more common in one gender or sex than another, such as the predominance of hair loss. The final pair, as I said, are the sex chromosomes. And as I mentioned, XX is female and XY is male. There's a couple of additional things that we can start to understand about the human karyotype, and one of those is the locations of certain types of genes. A very big project called the Human Genome Project has been used to try and map the human genome, that is, all of the genes that are present in humans. We found, for example, on chromosome 11, the gene for sickle cell disease, a particular disease that occurs more commonly in people of African nations, and one that gives them a little bit of an advantage over a disease known as malaria. We also know that on chromosome 7 is the gene for cystic fibrosis, and of course there are many more. So I have two questions for you really. The first is, what is the relationship between a gene and a chromosome? And what's the significance of the following? The relationship between a gene and a chromosome is that one individual chromosome can contain hundreds or thousands of genes. Genes are spread along chromosomes much like beads on a necklace, each individual one coding for a specific characteristic or protein. And one individual chromosome can therefore hold the information for a great library of different characteristics. When we look at the second of these questions, the significance of all of these letters, well, by now you should realize that these are actually the nitrogenous bases. Nitrogenous bases are the bases that are located between the sugar components of an individual nucleotide. A nucleotide consists of a sugar, a phosphate, and a base. And the base here is either a T, an A, a G, or a C. A sequence of nucleotides together, lots, is a polynucleotide. That is in fact what DNA is. So not only do we know that this is a particular sequence of bases along a chromosome or along a stretch of DNA, but we also could work out what the complement of each of these is on the opposite side, on the opposite strand of the DNA. Because we remember Tony Abbott and George Clooney, it becomes a simple matter of being able to identify which base is going to correspond with each of the ones listed above. How did you go?